So in our lab, we are interested in the membrane-associated signaling upon adhesion and entry, and also on pathogen adhesion to surfaces. So actually we are studying, for instance, the adhesion of Yersinia pseudotuberculosis to the host cells and the internalization. And you've may seen some membrane trafficking events. Also for Bordetella and Shigella. And just to please Jost, here you can see the recruitment of GAL3 on bacteria. So this is live imaging on host-pathogen interaction, and also studying their entry. But we are also interested in really understanding the adhesion process and the mechanical property of the membrane, because we are convinced that those mechanical properties of the host cell surface will organize the recruitment of signaling molecules. And to better investigate this issue, we are using biophysics. So we are using near-field microscopy, and for instance, we are studying adhesion of erythrocytes that are infected with Plasmodium, or also the interaction of Listeria with the host membrane, or Listeria biofilms to cell surfaces, or also spores with materials. And also, we are using atomic force microscopy to study at the nanometer scale the adhesion of pathogens or adhesin or bacterial toxins to the host cells. So we are really developing an interdisciplinary approach in the lab, mixing cellular molecular biology, microbiology, biochemistry, and biophysics. And as Philippe was saying, today I will focus my presentation on giving you a flavor of what you could do using this atomic force microscopy to study host pathogen interactions. So first I will introduce the instrument. So this is the AFM that we are using to study host pathogen interactions. So it is on an S2 facility. Here you see the laser of the TIRF system. It's an inverted here microscope. Here you have the optic fiber. Of course, you have an incubation chamber. And here you see the screens where we are following everything concerning the atomic force microscopy. And here all this, all the analysis about imaging, fluorescence imaging. Here you see the head of the atomic force microscope, and here the TIRF system. And this is the head system, and here you might see the cantilever, but here you will see it better. So this is the cantilever, and here is the tip of the cantilever. So the principle of AFM is quite easy to understand. You are following the deflection of the cantilever using the laser beam here that you follow you detect on the photodiode detector. So one first use of atomic force microscopy is to do imaging. I will give you, during this presentation, several topics that we are studying in the lab, just to illustrate the use of this technology. So for imaging, I selected red blood cells infected by Plasmodium, and we've heard a lot of talks this morning concerning this issue, and you may have heard during these talks about knobs, and this is the structures that we will follow here on these infected erythrocytes. These are the marrow clefts here. And to study here the knobs that are involved in cytoadherence of these erythrocytes to the blood vessel wall, we are using atomic force microscopy. Why? Because we can have here a very high resolution, nanometer scale. Here, the height is around 5 to 10 nanometer. This is extremely difficult to do with fluorescence. You will have to use super resolution techniques. But here with AFM, it's really durable. So thanks to these techniques, we can follow the structure of those of those knobs at different stages of development, and also using mutant strains of Plasmodium. And in all the cases, what we are doing is following these structures. And you can see here that these structures are really different. And here is the height, the height in nanometer. So using these techniques, we were able, studying different mutants, to make link between phenotypes and genotypes. But I won't enter into the detail of this story. What you can also do is to couple this technique with fluorescence, let's say biophotonic microscopy. And here, as an example, here you have cells that were stained with different markers, and you can do on top AFM scanning. So the interest of using atomic force microscopy to perform imaging is rather limited, I would say. What you can gain is information about the height with nanometer scale resolution. This is the main, to my point of view, the main advantage of using atomic force microscopy for this kind of study. The main advantage is really to study force interactions. Here you have the piezo movement of the samples that will get into contact with the cantilever. You will see that the cantilever will be deflected, and you will follow here the deflection using this laser. And here on this extension curve, you can extract a Young modulus that gives you information about the elasticity of the structure. And here, if you have interaction between a protein on the tip and a receptor, let's say, here you have an unbinding event, the disruption of this interaction. This is like a signature that you've got an interaction. So using this mode, which is called contact mode, then you can gain a lot of information that I will illustrate on the following slide. So here what we have two cells. In these cells, we overexpressed GFP protein coupled to anchor to the glycosyl phosphatidyl inositol moiety. And on the tip, we've coated aerolysine. Aerolysine is a bacterial toxin that recognizes these molecules. So here in fluorescence, you see that here we've overexpressed this protein into these cells. Here you see maybe the shadow of the cantilever. Here is the scan frame in the AFM. And now what we will get as information will be first the topology with this nanometer resolution in the z-axis. Then we will get information about the stiffness of the sample. And since we've coated on the tip this toxin that will recognize those proteins, we can map the distribution of these proteins at the cell surface. And of course, here we have much more because here we overexpress the protein. But on top of that, we can also really obtain the interaction forces. We are not talking about affinity here. We are really talking about forces and the interaction between the aerolysine and the GPI, GFP chimera. So with one scan frame, you get the number of receptors and their distribution, the interaction forces, the stiffness of the structure, and the topology, which is quite a lot of information that you can use on top of the imaging study that you are performing in the lab using confocal or video microscopy. So this is just to give you a relator. So here what we have is an axon from an hippocampal neuron. And here what you have, so it's 2 micrometer, the scale here. So you have the topology here, and all the pseudo-color code for the stiffness of the axon. And here the red arrows indicate where we've got interaction between GPI proteins and aerolysine. So following this information, then you can know whether you have interaction more on stiff domains or soft domains. And this is interesting if you are studying membrane heterogeneity on the cell surface. I will come to this point in 2 slides. First, let me introduce the way we are monitoring the stiffness of a membrane in a living cell. So what I said before is that you can obtain information about the stiffness of the sample when the cantilever is indenting into the cell. Now, what I will explain is that we are able, inside the cell, to really monitor only the stiffness of the membrane. And for this, what we are doing is what we call the relative stiffness. This means that for each pixel, what we are measuring and calculating is the Young modulus, let's say the elasticity of the structure compared to the elasticity of the surrounding membrane, if you wish. So that is to say, comparing the elasticity on the red square to the elasticity on the blue square. Oh, let's forget about this. So this is what this kind of scan frames that you can get. And then there is a lot of statistics, of course, behind to get the relative stiffness for a sample. And this is done, of course, on living samples. So as a proof of concept, we can talk about a raft. They have been introduced already today, but I will just add 2 lines of information. First, they are enriched in cholesterol, and if you extract this cholesterol, then you will perturb the structure of those domains, and you can extract the cholesterol using a drug that is called methyl beta-cyclodextrin. And now, if you follow the stiffness of the domain in which you have these GPI anchor proteins in function of time, you see that you are always about 100. And 100 means 100%, and this means that there is no difference between the pieces of membrane into which you have the receptor and the surrounding membrane. But since you see that all the points are above 100, this means that all these pieces of membrane, they are stiffer than the surrounding membrane. This means that the GPI domains are stiffer than the surrounding membrane. And here, we've added this drug, destroyed this structure, and you see that you have a drop, and you don't see this difference anymore. And this is true at 1 pixel distance, 2 pixel distance, or 3 pixel distance. So after destroying this raft, there is no differences in stiffness between these pieces of membrane into which you have these GPI anchor proteins and the rest of the membranes. Maybe it's easier to understand here, where here you have the cursor here indicates the stiffness. Here, this means that there is no difference between this pixel where you have the protein. You have to imagine that here you will have the cantilever, and here the aerolysine binding the GPI anchor protein, and here is the surrounding membrane. So the Z-scan is not height, it's stiffness. And minus 30 minutes means that it is minus 30 minutes before adding the drug. And here you see that we are always stiffer, and as soon as we will add the drug, then there is no difference anymore. This is not a model. This is coming from real data. So this is one way of using atomic force microscopy. Another way is to use AFM to study membrane trafficking. So it can be strange to hear, but actually, since you are following receptor on the cell surface, if those receptors are internalized, then you should be able to detect this internalization, meaning membrane trafficking. And in this case, we also used hippocampal neurons that were expressing a glutamate receptors coupled to an HA tag. And on the tip, we've put an antibody against this HA tag. And we did more or less the same experiment as I mentioned before. So here, what we know is that when we stimulate a neuron with NMDA, those AMPA receptors, they are internalized. And this is what we will follow here. Here is the control situation in black. And in red, here we have stimulated the neurons with NMDA. And what you see is a drop in the number of receptors detected, meaning that they are internalized. And the neurons, if you grab the spontaneous activity using tetrodotoxins, we know by biochemistry that those receptors that were internalized, they are recycled back to the plasma membrane. And this is exactly what you follow here. All these receptors, they are recycling back to the plasma membrane. And now if you do exactly the same thing, but analyzing the elasticity of the plasma membrane, what you see is more or less the same curve in red. And what you see is that all those receptors that were in stiff domains, they were internalized into the cells. And this is something that is also studied by biochemistry in neurons, is that these receptors, they are localized in stiff domains. Actually, they are localized into rafts. So this is illustrating that using the same strategy as before, you can also follow kind of membrane trafficking at the cell surface. Another example is coming from Listeria. Actually, the story started in Pascal's lab, in Pascal Kosser's lab, with Serge and Javier, Serge Mostowi. And what they found is what Javier was just mentioning at some point in his talk, is that septins are playing a role into the internalization process of Listeria. What they found is that expressing siRNA against different kind of septins, they could show on uninfected cells a difference in morphology that you can see here and here following actin staining. But also what they found is that Listeria invasion was impaired on cells that were treated with different siRNA, and they could reproduce this result using beads that were also introduced by Javier, beads coated by internalin bead. And what we decided to do is, based on their results, to study the interaction of internalin B with the MET receptor using atomic force microscopy. So, of course, with AFM, we could also follow the morphology of the cells. And here you see these differences in morphology, and here the differences on the height of the cells. But coupling on the cantilever, the tip of the cantilever, internalin B, we were able then to follow the interaction between internalin B and the MET receptor. Here you see all these signature for unbinding of the interaction. And we did more or less the same analysis as I mentioned before, but here what we were able to follow is really the interaction forces. And here you have the control situation. And this is an histogram of the forces and the number of unbinding events we could record. And what you see is that we could fit these results with two Gaussian curves that are also depicted here on the dotted lines here. But on these cells, we've got a shift for these interaction forces. This means that using AFM, we were able to detect differences in the force of interaction between internalin B and the MET receptor on these cells that were treated with this siRNA. Of course, we could also follow the topology and the stiffness of these membrane domains, and this is something that we are currently studying. I told you that we were able to follow the elasticity of the plasma membrane on the cell, on the living cells. But this we can do for the cell surface, but also inside the cells, because the cantilever is indenting into the cell. So the cantilever can sense inside the cells the difference in elasticity. And this is something that we've introduced, is kind of stiffness tomography. So here what you have is a fibroblast. Here is the nucleus that we removed for showing these results. And the yellow has no information. It's just to give you the images of the cell surface. But what you can see here is differences here in colors that reflect differences in elasticity, in stiffness inside the cell. So now the whole question is to know whether those corresponds to ER, cytoskeleton, endosomes, and so on. So we are currently correlating all these data with imaging in living cells with markers of each of those compartments and element of the cytoskeleton. So this is also something that can be really interesting. For instance, what we are doing now is using pathogens with actin tail and detecting differences in stiffness because, of course, actin is a structure that is easy to follow. So I would like to finish by giving you a flavor of what you could do with an IFM without using it as an IFM. This means that what we are doing is we are using an IFM as a nanomanipulator for fluorescence measurements, and you will see the advantage. Here, maybe you see a cell, and this cell express these GPI, GFP constructs that I mentioned before. And on this cantilever, we've coated some bacteria. So in that case, it is Yersinia. You will see them because they were labeled with DAP. And what we will do is that we will see here, so the bacteria on the tip, and now we'll put the tip on the cell that express this construct. And here what we are following is this construct. And what you see is the recruitment of this construct around the bacteria. And if you are following the complete movie, it's quite long, what you will see is different stages. This allows you to select the cell of interest, the cell that expresses a lot your marker of interest, and also to synchronize, of course, the recording. So when we engaged the cantilever and the tip onto the host cells, what we saw first is a recruitment of all these molecules around the bacteria. But the bacteria, they are fixed on the cantilever. They can't be internalized. Okay, they are really stick on the cantilever. So what we saw, because on this tip we had actually 3 kinds of bacteria. So these ones, those ones, and all those were living bacteria. And here you've got a dead bacteria that we killed before putting on the cantilever. And what you see here is that all this labeling is now shifting from this bacteria to the next one here. And you can imagine that it is because this one they can't be internalized. But a big advantage of using AFM, for instance, versus optical tweezer, is that here you can retract the cantilever. The bacteria, they will stick on the tip. And now you can move your tip to go here. So you move the tip, and then you re-engage. And what you will see is that the proteins, they are recruited once more around the living bacteria. So this technique allows you to do multiple retraction possible, which is not the case with optical tweezer. And the last example that I wanted to share with you is this one. And actually, here what we have is, again, a Yersinia on the tip. And then we will put this Yersinia onto the cell, okay? And now using TIRF technique, we will follow the movement of the cytoskeleton near the substratum. So we will just play with the bacteria to do this and look at the actin cables, how they will react when you play with the bacteria on top of the cell. And for this, so we use TIRF, and we selected this actin cable here. So this is a TIRF image. Here is the bacteria. Of course, so here we will follow the fluorescence. Here it's the fluorescence of the bacteria, and here is the fluorescence of the actin cable on two separate channels that are combined after that. So here what you see is that the tip is engaged and the bacteria is touching the cell. And now, if you look at the actin cytoskeleton, then you see that there are some, let's say, polymerization going on just below the bacteria that you can follow in function of time. So here. So this is just to illustrate that you can combine now AFM and other techniques, like, of course, you could do it with confocal, although I would not recommend it because for simultaneous measurements, it's really, really hard. But for instance, TIRF, it's really doable. And now what we will do is to go and use other technique that I will mention now. So you can also use FRET, FRAP, or FCS. FCS also is like confocal. It's really, really difficult for obtaining simultaneous recording. But something that you can do is to couple this technique with super resolution techniques, let's say state, storm, palm, and so on. What is the interest? Those techniques are fluorescence techniques that allows you to obtain as a resolution, let's say 20 nanometer on living cells. And this is exactly the resolution we obtain with AFM on living cells. So this means that now we have two techniques that are speaking the same language with the same resolution. So this is something that now we are developing in the lab. So in conclusion, I would say that AFM is a mature technology to address mechanical issues, but also host pathogen interaction issues with high resolution on living cells, and that is available for cellular microbiologists, if you wish. It always still some possibilities of fast scanning because the resolution, the temporal resolution is, let's say, okay, for a scan, 5 minutes. So to obtain nice data on the, let's say, a scan which is 2 micron per 2 micron. So it's quite a slow technique. So there are people working on this issue, especially here in Paris, for instance, at the Curie Institute. And for us, we have decided to go for the simultaneous combination with high resolution imaging techniques. So we prefer to privilege the spatial resolution. So with that, I will thank the people involved in the AFM technique in the lab. So it's Sébastien Janel and Yann Sexora. And among the collaboration for the result I presented, so Michel Simonet for Yersinia, Pascal Cossard's group, and for Listeria, Denis Mattei for Plasmodium. For all the AFM is done in collaboration with people from the EPFL in Lausanne, at the Complex Matter Institute, and also the Physics Theory Institute, and also for the programming for the softwares with Charles Roduit at Humble. And these are for the financial support. And with that, I will thank you for your attention.
Merci beaucoup. Thank you very much, Franck. Any questions or suggestions or requests for Frank's phone number if you want to collaborate with him?
So thank you very much for this nice talk. I just have a question because I think that atomic force microscopy have been used for visualizing nucleosome structure. Particularly, Stefan Dimitrov in Grenoble is doing that kind of experiment. So do you think that the technology now is mature for visualizing some chromatin state in living cells
It depends. If you are talking about imaging, then I will recommend to use fluorescence. If you want to obtain biophysical properties, then, for instance, for chromatin, if you want to know if you have, although I don't know if it's possible, but differences in stiffness in the nucleus, for instance, this you can try, although I guess it is really, really difficult. As you've seen, we removed the nucleus because it's really a stiff structure. So we had to change our settings to study nucleus. It's possible, but it is really, really difficult. But it's possible. There are people doing that, or at least trying.